Hello, Science 30, and welcome to Cell Division and Interphase. This is our first lesson in the molecular genetics section of our bio unit. We are going to start off our lesson by looking at some key vocabulary. So today we're going to learn the role of the cell cycle and understand its key vocabulary. We're going to look at the different parts of the cell cycle. We're going to create a pie chart and we're going to explain what's happening in that pie chart. And then we're going to use the different vocabulary words as we go throughout the course, becoming more comfortable with them. So in lesson 5, we are going to start off with the terms parent cell and daughter cell. Now, this isn't the first term you have on your vocabulary sheet, but we are going to define them first. When I use the terminology parent cell, what I'm talking about is the original cell that we started with. When I use the terminology daughter cell, what I mean is that parent cell has split into either 2 or 4 cells, and those become the daughter cells. So they're the newly created cells through division. Please make sure to copy each of these definitions directly into the definition portion of your vocabulary piece. So here's an example of a parent cell, one cell, and if I divide that parent cell out, that's a daughter cell. Just a little visual here to help you understand the terminology a bit more. Now, we are going to focus on chromosomes. We are going to be looking at chromosomes throughout this chapter. And what a chromosome is it is a structure made up of condensed DNA. It can be individual or replicated, and it contains what we call sister chromatids. I only just need you to write that it's made up of condensed DNA. All right. Now, there are a variety of different ways we can look at a chromosome. A chromosome could be a single structure like this. That is the individual version that this definition is referring to. Or it can be the duplicated version or replicated version, which would be what you see here. And these contain sister chromatids. So what do we mean by condensed DNA? DNA looks like this inside cells. It looks like a giant plate of spaghetti. And when we need to actually divide cells out through the cell cycle, we cannot just have this giant plate of spaghetti, or chromatin is what it's called, floating around. Instead, we need to condense it down. So that's what a chromosome is just condensed DNA. Another very important terminology you're going to need to know as we go through this is the word diploid. Diploid is represented by this right here, 2N. 2 being double the amount of the DNA we have, so 2N. So we say this is pairs of homologous chromosomes, which just means that we put 2 chromosomes together. We typically talk about a diploid organism being something that is within our body cells. So all of your body cells are diploid. That means you have 46 chromosomes in them because you have the full set. Now, diploid also can be contrasted with haploid. Haploid is looking at unpaired chromosomes. So instead of having them paired up, they're all on their own. If a full set of chromosomes is found within all your body cells, then that means your half sets are found within your sex cells. How you would know how many are in a haploid organism is you would divide their diploid number by 2. So 46 are in humans. If we divide 46 by 2, so if we divide this by 2, our N, what we're left with is 23. Okay, so haploid is 23 for humans. So what you're going to do is you're going to write down the definition, and then we are going to talk about diploid and haploid all the way through because different cells we look at will have different diploid numbers and different haploid numbers. That now brings us to the whole reason why we're talking about this terminology, which is cell division and the cell cycle. So the cell cycle, or the life cycle of a cell, is what happens to all cells that lives in the somatic body cells, and it's based off of the environment that they're in. Okay, your blood cells, which we've learned about, skin cells, are actually replaced quite frequently. They divide often, okay, but nerve cells do not. So when we talk about the life cycle of the cell, or the cell cycle, we're really just talking about the dependency of how quickly they divide, and not all cells divide quickly. So a single cell cycle is defined as a sequence of the event from one cell division to the next. And we're going to talk about cell division as mitosis and meiosis. So we'll get to that at a later lesson. There are generally 2 phases that we focus on in the cell cycle. One is interphase, which is the lead up to cell division. And then the second part of it is cell division. In interphase, you have three distinct phases that occur. G1, which is cells grow. So for your notes, just put in cells grow. That's all you really need to know. In S phase, we're going to focus on DNA replication here shortly, but in S phase, DNA replication occurs, meaning we're going to duplicate the amount of chromosomes we have so we can pull our cells apart and have consistent amount of chromosomes in each. And then G2 is where we continue protein synthesis and get ready for division. So we gain more energy, we grow a little bit more, and then we divide. So these three parts of interphase happen, and each one happens with a checkpoint. So G1, cell grows, checkpoint. Yes, we're good, growing fine. Let's replicate our DNA. Once DNA replication has occurred, check, we're done that. We're going to move into growth again. So there's checkpoints all along for cell division to occur. What we're going to do now is we're going to go into the little box that says draw a simple diagram of the cell cycle. So here's what it looks like. It looks like a little pie chart. And what we're trying to represent is the length of time that the cell spends in each phase. So the first thing you have to remember is that interphase consists of three distinct pieces. So I've shown you that interphase is this part of the cell cycle, this part, and this part. Then you have cell division. Cell division and interphase are not the same process, so we separate them out. The first part of interphase, again, is that G1, where we see cell growth and metabolic activity happening. And it is the longest time that the cell spends in interphase is in G1. Then we have S phase, where we have DNA replication occurring. So again, that's just when we double our DNA. And then lastly, we have the cells prepare to divide, so it grows a little bit more and gains energy before it goes through cell division. A cell will spend most of its lifetime in interphase before it goes through either mitosis or meiosis in cell division. So this little representation in your notes is just to give you a visual of how the cell cycle works. If you have any questions, please let me know. Please make sure to have all your definitions and your pictures drawn, and I will see you next time.